All right. Hi, everyone. It's 10:30, so we're going to get started. For the next few minutes, I'll just review some webinar protocol and kind of review what the Phragmites Collaborative does. So Megan will start in about 5 to 10 minutes. So as people keep coming in, I'm going to start on my little spiel. If you've been to one of our webinars before, you've definitely heard it. But welcome everyone, and this is, I think, the Great Lakes Phragmites Collaboratives. We're now on our 51st webinar, which is very impressive. I'm Teresa, and I'm joined by my colleagues James here, and we're hosting today's webinar on behalf of the Great Lakes Phragmites Collaborative. And our presenter today is Megan Jordan, and she's from the University of Waterloo, and she will be presenting native wetland plant recovery following herbicide treatment of Phragmites australis. As I mentioned before we get to her presentation, I have a few housekeeping items that I'm just going to briefly go over. For those who may not be familiar, I just wanted to introduce the Phragmites Collaborative. I'm sure most of you are familiar, but the Phragmites Collaborative is a partnership to link people, information, and action against invasive Phragmites. The collaborative is coordinated jointly by the Great Lakes Commission and the USGS Great Lakes Science Center with funding from the Great Lakes Restoration Initiative. And the collaborative facilitates communication among stakeholders across the region and assists in development and management of tools in a program to serve as a resource center for information on Phragmites. The collaborative is led by a multi-agency advisory committee and applies principles of a collective impact to advance a common agenda for Phragmites management in the Great Lakes. And here's our common agenda. The Great Lakes Phragmites Collaborative has been working towards advancing member strategies that are outlined here in our common agenda. And this is one big step of aligning regional goals and establishing a shared vision for change. So the purpose of our common agenda is to clearly define the intentions of the collaborative, develop a shared understanding of gaps and problem areas surrounding Phragmites, and to highlight areas and strategies that we are working on or wish to work on in the future. And you can learn more about our regional efforts and how to get involved by visiting our website, which is here on the screen. A big component of our work and one of the ways we engage with Phragmites managers the most is through the Phragmites Adaptive Management Framework, or as we call it, PAMF. PAMF utilizes participatory science and provides data-driven Phragmites management guidance through an adaptive management framework. Basically, this just means we've established a way to learn what Phragmites managers are doing out in the field and turn that data into annual guidance to help managers make effective and efficient management decisions. You can learn more about this program at our website. The URL is on the screen. Or if you have any questions, you're welcome to email our PAMF coordinator, and their email is pamf@glc.org. But if you or anyone in your agency are interested in joining PAMF or taking advantage of PAMF's guidance, you can enroll on your own terms. We have free virtual training courses, which you can access by following this URL, and lots of ways to help you get started throughout the year. As mentioned, you can email pamf@glc.org if you have any questions of how you could benefit from this program. And if you're interested in learning about more Phragmites management research, our Great Lakes Phragmites website has a ton of resources, including management guidance, and we have all of the recordings of our past webinars. You can join our listserv to connect with hundreds of other professionals. We have a bi-weekly newsletter, which goes out, and a quarterly research roundup of academic papers published in the previous months. And you can join all these resources, the listserv, the newsletter, by going to our website, which is here on the screen. Additionally, today's webinar is going to be recorded and will be uploaded to our YouTube channel and posted on our website. You can access all of our past presentations on our website. There you can view past webinars and also register for upcoming webinars. And of course, if you want, you can connect with us on Twitter and Instagram @GLFrag to stay connected. Okay, that's my little spiel about the collaborative. We're going to go over some logistical housekeeping for the presentation. But in order to minimize interruptions to the speaker and presentation, we We ask that you stay muted and your cameras off through the remainder of the webinar. We encourage questions throughout any time you have questions, and they may be submitted through this chat box, which is in the green circle here. So please, as you have questions, as Megan's presenting, submit them to the chat box. We will go through the questions at the end of the webinar in a Q&A portion. If you're having any technical issues with Zoom or any of the connection information, you can also send us a message through the chat. You can direct the message directly to the host, which will bring the message to me or my colleague James, and we'll do our best to try to answer them. Okay, that was my spiel. So now it's my pleasure to introduce today's presenter. So Megan is a research assistant at Dr. Rebecca Rooney's Waterloo Wetland Lab at the University of Waterloo. Megan completed her master's in the Waterloo Wetland Lab in May 2022, in which she examined native wetland plant recovery in Lake Erie coastal wetlands following herbicide treatment of Phragmites australis. Following the completion of her master's degree, she continues to monitor these vegetation communities that she's grown fond of and has now started to examine the indirect effects of herbicide treatment on the wetland as a whole, such as the effects of waterfowl foraging and others. Outside of the field season, Megan loves volunteering for dog rescues, crocheting, and cross-stitching. Sure, great hobbies. So thank you so much for sharing your work with us today, Megan. Feel free to bring up your slides and take it away.
Okay, awesome. Like I was saying, I'm using Canva, so hopefully this all works. I will present. Okay.
It looks
Is
good.
it good to go? Awesome. Yep. Okay, I will minimize that screen there. So hi everyone, thank you for coming to my presentation today. It's super exciting for me to present today. I'll be talking about native wetland plant recovery following Phragmites australis invasion, which is my thesis topic. I did defend my thesis, like Teresa said, in May of 2022. And this is kind of the first time that I'm getting to talk about my thesis work since defending. So super exciting for me. So thank you again for all coming. And we'll get started. There we go. So before we get started, I just want to thank Environment and Climate Change Canada, the Canadian Wildlife Services, and the Ontario Ministry of Natural Resources and Forestry for all of their funding, as well as the tireless volunteers who helped me with all of my field work and thereafter. I really couldn't have done it without each of them. Oh, I wasn't nervous, now I'm nervous and now I'm stuttering. But and of course the rest of the Waterloo Wetland Lab for all of their support. And just a little shameless plug before I start, we are looking for a summer wetland field tech. If you know anyone in Southern Ontario who might be interested, please get in touch. This whole posting can be found on my supervisor, Rebecca Rooney's website and Twitter account if you're interested. So Phragmites australis, I know who I'm talking to today. I won't go into too much of a background or an introduction about Phragmites, but if you are not already aware, it is a tall invasive grass in North America, and it negatively affects many native plant and animal species, particularly with plants. It outcompetes them by shading and overcrowding them, and this creates kind of a monoculture of Phragmites where it grows. It reproduces primarily vegetatively via rhizomes and stolons. However, it also produces lightweight seeds that have been shown to be a viable method of reproduction. My study sites were along the northern shore of Lake Erie at Rondo, which is here on the left, and at Long Point, which is here on the right. These two wetland complexes have experienced a large-scale invasion of Phragmites australis since the mid-1990s, which is believed to have been facilitated by a period of low water levels. To date, over 1,500 hectares of invaded marsh has been treated with a glyphosate-based herbicide since 2016, and this herbicide was applied via aerial and ground application methods, so aerial via helicopter or ground via Marsh master. And despite the wealth of research that has gone into Phragmites, there are still many knowledge gaps that remain. Firstly, there is a lack of long-term studies in Phragmites research, and this is particularly important because we don't necessarily want to know what happens 1 or 2 years following herbicide application or invasion. We really want to know the long-term effects of invasion or herbicide application. We also don't necessarily know how effective the ground and aerial application methods are relative to one another, nor do we know if herbicide application will affect the wetland seed bank, and if it does, if Phragmites will return via viable seeds following treatment. So that brings me to my first experiment, the herbicide efficacy experiment. My objectives for this experiment were firstly to determine the trajectory of Lake Erie vegetation communities over a 6-year period following herbicide application, and also to compare the relative efficacies of the ground and aerial application methods. So to do this, we established a total of 120 permanent monitoring plots in Rondo and Long Point in a before-after control design. 40 of these sites were control that were in areas of highly dense Phragmites that remained untreated throughout the duration of the experiment, except unfortunately for us, though they were treated in 2020, and so we don't have data for them for 2021 or 2022. However, we also have 40 treated sites that were treated in 2016 via aerial application, and 40 sites that were treated in 2017, 20 of which were ground treated and 20 of which were aerially treated. Myself and my lab monitor these plots yearly between 2016 and 2022, and hopefully we will continue to monitor them in the upcoming years. So for each of these surveys, we monitored the water depth, the canopy height, the live and total Phragmites stem density, the incident light reaching the water surface, as well as the percent plant cover in each of the 1 meter squared quadrats each year. So starting with the result that is perhaps the most evident, Phragmites suppression. So in each of these figures that I'm going to show you in 2016, that's 1 season prior to herbicide application. So the treated and control plots should look very similar in each of the figures. The real differences should be seen after 2016, so 2017 to 2022. And you'll see the differences between the gray circles, which are the control plots that remained untreated until 2021. And the green triangles, which are the treated sites. So, like I said, starting with Phragmites suppression, as you can see in 2016, the sites look very similar between treated and control. Following herbicide application in 2016, we really see a decrease in Phragmites stem density, which is really great news because that just shows that the herbicide application was really effective in reducing the number of live stems and over time reducing the number of total stems. So as we know, Phragmites stems can remain standing for a couple of years following herbicide application, but we did see a decrease in total stem density over the years as well. Very similar to this trend seen here. So in addition to a decrease in Phragmites stem density, we also saw a decrease in canopy height and an increase in light penetration. This is really great news for the native seedlings that are trying to emerge following herbicide application because they really need that light penetration to germinate and grow. So now moving on to richness and diversity indices. These trends are a bit different than the Phragmites suppression trends. So we'll start with the first 2 years following herbicide application here. We actually see a decrease in species richness, as well as Shannon-Wiener diversity, Simpson's 1/D, and Pelu's evenness. This is mainly due to a round of secondary invasions that we experienced in many of the plots following herbicide application, mainly due to European frog bit in the first couple of years following herbicide treatment. Many of the plots were really choked out by European frog bit. Unfortunately, this is quite common in Phragmites treatment projects because the herbicide treats not only Phragmites, but all plants. And so once you kind of have that blank slate, it's really easy for secondary invaders to come in and kind of rule the roost. But moving on to the good news, in the next 4 years or so following that secondary invasion, we have seen an increase in species richness, Shannon-Wiener diversity, Simpson's diversity, Pelew's evenness, and mean coefficient of conservatism. This is due to that secondary wave of invasives kind of dying off, and we see a reinvasion or a reintroduction of those native plant species and native plant communities coming in the next 4 years following those initial secondary invasions. So if you put all that data together, you kind of come up with this really, really pretty trend of the first year, of course, before herbicide application took place. All of the plots were Phragmites dominated. Then we see the secondary invaders coming in and dominating those plots in the first 1 to 3 years following herbicide application. And then kind of, like I said, beautifully symmetrically, you see an increase in the native dominated plots to the point where in 2022, around 90% of those plots were native dominated. So now moving on to the differences between aerial and ground application. We'll start with aerial just because it's a bit more straightforward. So like I said, first year following herbicide application, most of the plots were non-native dominated. And then we've seen a bit of a messy trend, but overall we see an increasing trend towards native dominated plots to the point where 5 years post-treatment, we see 70% of those plots were native dominated, and hopefully that trend will continue to increase in the future. One thing that I do want you to pay attention to is the fact that none of these plots were actually Phragmites dominated at all following herbicide application, which is very different in comparison to the ground applicated plots. As you can see in the first year following herbicide application for those ground treated plots, 40% were still Phragmites dominated, despite this being 1 year following herbicide application. That fell to 10% of plots in the second year following herbicide application. And then this kind of follows the other trends with the increasing native dominated plots throughout time. Now, the difference between these two different application methods is kind of up for debate. We hypothesize that perhaps it's because of when the Marsh master was treating those plots initially, we weren't using a fine enough filter to combine the water coming in from the marsh with the herbicide. And so maybe some of that muck was kind of decreasing the efficacy of the herbicide being applied in the Marsh master. Since then, we have increased the filter, or I guess decreased the filter size to make the herbicide more effective. And hopefully moving forward, we'll see a higher efficacy with those ground application methods. So in conclusion, secondary invasions may occur following herbicide application, but may also be short-lived, as shown by those secondary invasions by European frogbit. That native vegetation recovery following invasion is possible, that aerial application may be more effective than ground application, although that may change in the future with that decrease in filter size. And finally, that long-term monitoring projects are crucial, as shown by the long-term nature of the trajectory of the vegetation following herbicide application. So all of that experiment has taken place here shown in the purple area. So that was a time period where Lake Erie water levels were relatively high. But as many of us know, Lake Erie water levels are beginning to drop. And with dropping water levels, that leads to a change in vegetation communities. So now that we know what has returned when Lake Erie water levels are high, can we be proactive and determine what may return under falling water levels? Well, the answer is yes, and that was done by a Seedbank experiment, which is the second experiment in my thesis. If you don't know, the seed bank in a wetland consists primarily of the top 2 centimeters of sediment. This is where a lot of seeds kind of settle in the wetland. And from there, they can either perish due to old age or environmental conditions, or emerge and become a seedling. And seedlings emerge primarily under low water levels from the seed bank, mainly due to increased light and increased oxygen levels. And because of this, the seed bank can help determine what will emerge under these low water level conditions. So my research objectives for this experiment were firstly to determine the effects of herbicide application and flooding on the wetland seed bank, and next to examine the wetland seed bank to determine what may return following those falling water levels. So to do this, we collected seed bank samples from 60 sites at Long Point, 20 of which were control sites in areas of highly dense Phragmites australis, 20 of which were herbicide treated, so previously invaded by Phragmites australis, but treated with herbicide as part of the herbicide treatment program at Long Point. And finally, 20 of which were reference sites in areas that were never invaded by Phragmites and therefore not treated as part of the project. At each of these sites, we collected the top 2 centimeters of sediment, which is, like I said before, where the highest density of seeds kind of settle in a wetland. Back at the lab, we sieved each of these samples to remove any large stolons or rhizomes or tergians to ensure that any seedlings that emerged from the seeds themselves and not from vegetative methods. So at the University of Waterloo greenhouse, we exposed these seed bank samples to 2 different watering regimes. The first one was a flooded regime, where the water levels were maintained 2 to 4 centimeters above the soil surface. And then the other was a moist regime, where soil moisture was maintained but never flooded. We watered each of these samples for 7 months, every 1 to 2 days. And despite how laborious that sounds, it was actually probably my favorite part of my thesis work. It was really nice spending a lot of time in the greenhouse and kind of watching these seedlings emerge every day. And as you can see, it was quite successful. A lot of seedlings emerged. Here is just some of my favorites. So on the far left here, we have nodding water nymph. In the middle, stinging nettle. And this is something that I say during every presentation, and people make fun of me for it, but I'll say it again. I didn't have to ID this with a key. All I really had to do was let it sting me about 5 times, and that's when I really recognized that this was stinging nettle, so very easy to identify. And finally, purple loosestrife, which is, as we know, another invasive species in many Lake Erie wetlands. So in addition to those, we also had wintercress, which, although it's very pretty, is also another invasive species. We saw lots of European frogbit and, of course, tons of Phragmites. So this is what the abundance graph looks like. As you can see, it was very, very successful project. We found around 3,000 seedlings emerged in total. So first, starting off with Phragmites seed density, as you can see, perhaps the most obvious result is on the left here in the control group. I've separated each of the seedling densities into the flooded and the moist regimes just to make the results a bit more clear. And as you can see, the highest number of Phragmites seedlings emerged under the control moist condition. So that's probably to be expected, as where you have very dense Phragmites, you are going to have lots of viable Phragmites seeds in the seed bank. But if you compare it to the flooded condition, you can see that there is a huge decrease in the number of Phragmites seeds that emerged under the flooded condition, so under 2 to 4 centimeters of water compared to the moist condition. So that's really important, as it shows that just 2 to 4 centimeters of flooding is sufficient to suppress Phragmites emergence from the seed bank level. And then again, if you compare control to the treated sites, you can see that again, there is a huge decrease in the number of Phragmites seeds that emerged under the treated condition. So that's Phragmites that has been treated for at least 1 year. And then of course, because water levels are high, have been flooded for at least 1 year. We see that Phragmites seed viability was greatly reduced under those conditions in comparison to the control conditions. And then now looking at native seed density, these trends are a bit less clear. You can see a similar number of native seeds emerged under the control reference and treated conditions under the flooded and moist watering regimes. This is also great news as it shows that even under very dense Phragmites, under the control conditions, we see many native seeds, viable native seeds still present. So that just shows that perhaps if that Phragmites was treated, that those native seedlings might have a better chance and might be able to reestablish a very diverse native vegetation community following herbicide application. So the bad news is if you compare those y-axes, you might see that the number of Phragmites seeds that emerged compared to the number of native seeds that emerged might show that there are tons more Phragmites seeds in comparison to native seeds present. Now this isn't necessarily the end of the world, as it just shows that perhaps those treated areas might need to be actively seeded with native seed mixes to ensure that those native seeds have a bit greater chance of surviving following herbicide application. And just accounting for the other seedlings that I've not shown, the non-native seedling density is shown here. This includes things like purple loosestrife and cattail, which are largely invasive in Lake Erie wetlands. As you can see here, a lot of those seedlings emerged under the treated and control conditions. And this is just something also to keep in mind that when looking at non-native emergence, we're not necessarily just looking at Phragmites emergence. We also have to consider cattail and purple loosestrife and wintercress and things like that. So it's really important to keep in mind. So in conclusion, many viable Phragmites seeds remain in untreated marsh, so approximately 185 seedlings per meter squared. That herbicide treatment and flooding have decreased the density of viable Phragmites seeds. And finally, that active restoration, so maybe actively seeding the areas with native seed mixes, may be necessary to ensure native vegetation recovery. And despite all of these findings, there are some shortcomings to greenhouse experiments that I will acknowledge. The greenhouse is highly controlled in terms of temperature and elements, so there was no wind, there was no snow, there was no sleet or hail or anything like that. Things that those native or those seedlings may be exposed to in the wetland itself. There is also no competition as the seedlings were removed once they were identifiable, so there's no shading or priority effects. And there's also no herbivory, so that may drastically change the type of vegetation communities that persist throughout the growing season. We didn't see any of that in the greenhouse experiment. So that brings me to my final experiment, which takes all of those things into consideration, the marsh organ experiment. So a marsh organ, it might sound really weird. I know it sounded weird to me when I first heard it, and it also kind of looks a little weird, but this is a little schematic that I made of what a marsh organ is. The name marsh organ comes from pipe organ, because that's kind of what it looks like in the marsh. It consists of a bunch of different heights of pipes that are hammered into the marsh, and then you fill them with sediment, and then in the top, you place either a fully grown plant or a seed bank sample, and you leave them in the marsh throughout the duration of the growing season, maybe more than that. And then you can find out the effects of different water depths in situ. So how that works is these shorter pipes are exposed to a higher water level as the water comes up almost all the way to the top of the pipe. And then the taller pipes are exposed to a lower water level as the water only comes up partway through the pipe. So it's a bit confusing, and I know it's a bit weird to think about at first, but hopefully this diagram kind of clears things up. So like I said, it exposes plants to different water depths in situ in the wetland itself rather than in a greenhouse, for example. So my objectives for this experiment were firstly to examine the wetland seed bank to determine what may return following falling water levels. So the same as my last objective in my last experiment, and also to determine how Phragmites and other non-native seedling emergence changes under different water levels. And finally, to determine how emergence changes in situ, so in the wetland itself, as opposed to in a greenhouse. So for this experiment, we had 15 sites, 5 of which were control sites in areas of highly dense Phragmites australis, 5 sites which were treated in 2020, so were previously invaded by Phragmites australis, but were treated with the glyphosate-based herbicide, and 5 of which were reference sites in areas that were never invaded by Phragmites and therefore were never treated. We collected seed bank samples at each of these sites and combined them. So all of the control sites were combined to make the control seed bank and treated in reference seed banks. We also sieved these seed bank samples to again ensure that any seedlings that emerged from seeds and not from vegetative methods. So marsh organs construction, this is something that I still have nightmares about, and I'm not joking. I have about 2 nightmares about it a week still. But this is what my marsh organs look like. We had a total of 12 pipes total with 4 heights, so 3 pipes per height. The first depth was at 0 centimeters, so at the water surface, right here. The next one was 33 centimeters above the water surface, then 67 centimeters and 100 centimeters. We chose these depths due to the variations in Lake Erie water levels, as they tend to fluctuate by about a meter over the years. And so we really wanted to capture that variation in the marsh organ. We then hammered the pipes into the sediment, filled them almost to the top with sediment, with nearby sediment, and then placed the respective seed bank slurry into the top of each of the pipes. And luckily, despite all of my anxieties, each of them stood throughout the entire summer. All 15 marsh organs stayed standing, fortunately. This is just a picture of what one looked like in April 2021 when we installed them in the middle of the very, very cold spring. And this is what they looked like at the end of the growing season when I took them down in September of 2021. And here are some more pictures. As you can see, some of them, some of the marsh organs did a bit better than the other in terms of seedling emergence, also in terms of stability. As you can see, this one was the first one we made, and it's a bit topsy-turvy, but it stood, and it also produced some great seedlings. And then these 2 are actually both control sites, so they produced a ton of seedlings. They were also a bit more protected, which might explain why they might have been more productive than the first one. So emergence, we had some amazing plants emerge. I was so excited to get to see them every month or so that we went to monitor them. On the far left here is a poplar seedling, which was, I think, the most surprising thing that emerged from my marsh organs. We did find a dead bird in that pipe at one point. So I'm not sure if the seedling emerged from the seed bank or from the contents of its stomach, but it was very cool either way. And then in the middle here, we have arrowhead in one of the 0 centimeter pipes. And of course, my nemesis, stinging nettle, got stung by it some more in the Marsh organ pipes. And then here on the far left, we have more wintercress, again, another invasive species. Bittersweet nightshade in the middle there, another invasive species. And of course, on the far right here, lots of Phragmites and lots of pale loosestrife, which is another invasive species, unfortunately. So unfortunately, I wasn't able to include this data in my thesis. However, I'm still hoping to publish it very soon once we are able to analyze the data. We really want to look at how Phragmites emergence, non-native species emergence, native species emergence, and community composition differ between vegetation types and, of course, between the water depths in the different heights of pipes. We also, like I mentioned before, would love to see how seedling emergence and survival changes under active restoration, so actively seeding treated areas with high and low diversity seed mixes and high and low density seed mixes. And luckily, one of our members of our lab, Jersey, is actually looking at this. We're going out into the field in a couple months to actually look at this and try to answer these questions, which is very exciting. Jersey's email is here on the screen. If you would like to get into contact with her, feel free to email her or email me and I can forward you along. And this is Jersey in this picture here. As you can see, my supervisor, Rebecca, somehow managed to get me to do marsh organs another year. I don't know how she managed to do it, but we did it again last year. So Jersey will be having a lot of really cool data to look at in her thesis. And I'm really interested in looking at the results. So that is all. Thank you so much. I'm happy to take any questions, but I'm also happy to answer any emails or connect with you on Twitter. I'm not super active on there, but it's another way to connect. And also feel free to check out our lab website as well. The job posting is also on there. And also you'll be able to connect with a bunch of my awesome lab mates as well. So that is all. Thank you so much.
Oh, thank you, Megan. That was so great. I already see there's a lot of questions in the chat, so I hope you're ready.
I saw them popping up during the presentation.
Hopefully that wasn't distracting while you're talking.
No, it's okay.
I normally put up like a cute thank you slide, but actually I'll let you leave up your presentation in case you want to click back to anything.
Okay.
So I'm just gonna start. I'll start at first. So these will be probably like about some of your first slides.
Yeah.
A question from Diana. What herbicide was used for the treatments?
It's a glyphosate-based herbicide. I haven't totally memorized all of the loading rate and everything, but it's a glyphosate-based herbicide.
Okay. And from Lisa, what time of year did you treat?
It was treated in the fall, so late September, early October, depending on like the year and how weather conditions were and everything.
Also, this is to everyone. You guys can keep putting questions in the chat, and we'll keep reading them as long as we have time. So, from Taja, do you have any hypotheses about why the frog bite actually started to decrease? Was it actively managed in the area? Do you know?
It wasn't actively managed, which is why it's still kind of a mystery as to like why it came and left. We were kind of talking about it when I was writing my thesis, and I think my hypothesis is because of the water levels, just because they were so high for a while and then they've kind of been decreasing. So I think they definitely had an influence on the kind of vegetation that was there and kind of like what persisted and what didn't.
That's super interesting though. And then another question, did the frog bit naturally decline? So maybe, yes.
Yes. Yeah.
I think this is for the first part of your presentation. A question from Lynn, what were the other non-native species that dominate the plots besides frogbit?
Yeah, so tons of frogbit, like mats of frogbit the first couple years. We also saw Eurasian milfoil, which has kind of been making a resurgence in many of the plots in the last couple years. Potamogeton crispus, curly-leaf pondweed, purple loosestrife, just kind of like the big invasive species that are quite common in Lake Erie wetlands.
All the big players there.
Yeah.
Was the frog, this is from Katie, was the frog cut as well or left standing? And they're referring to both the aerial and ground treatment.
Yeah, so in Long Point, it was rolled. At Rondo, they were trying to do a prescribed burn, but the weather conditions and the wind never really lined up to like be able to do that. So there was no secondary treatment at Rondo, but there was at Long Point.
For a question from Lynn, do you anticipate that lower water levels would start to bring up the frog invasion again in this area?
Yeah, so
looks like yes from the seeds.
Yes, unfortunately, but hopefully in those treated areas that have been like treated for a couple of years now, that the flooding has and time have kind of decreased the viability as well as the presence of those native seeds. Hopefully they can stand a chance and kind of outcompete that frag if we do the active restoration and active seeding. Hopefully that'll increase their chances. But yeah, I think it's kind of a ticking time bomb with water levels decreasing and having so much frag seed in the seed bank.
Another frogbit question. Do you know, was frogbit observed prior to the Phragmites control, or did you guys did you see it in the wetlands around the area, or did it just seem to emerge following the control?
It was present before treatment took place, just not in as high densities as it was after treatment took place.
Just a comment that stinging nettle is great to eat. Thanks.
Oh,
I've never had
yeah.
it.
I think someone did tell me that when I was doing it in the greenhouse, but yeah.
I'm sure you have to prepare it a certain way. This seems to be, I don't know if you'll know it, probably everyone wants to know. A question from Joelle: how long will Phragmites seeds stay dormant underwater? Do they seem to die when they're fully saturated?
So just based on the results from my greenhouse experiment, it looks like 1 year of flooding and time is enough to kind of like decrease the viability, not to zero, but in that figure, I'll go back to it. It really, there we go. It really decreased in the treated, the Phragmites seedling density really decreased in the treated sites even after 1 year of flooding. So hopefully that's enough to kind of keep them at bay.
Awesome. A question from Lynn: does that, I'll have to maybe read it and you'll know what they're referring to. Does that mean that the treated sites are also affect seed viability of the seed bank?
Not totally sure what Lynn is referring to there.
You can pop on if you want to clarify your question, if you're still here.
I don't type very well.
It's okay.
I was just wondering if the Phragmites seed bank, like when you treat, are you killing all the seed bank? Like, did you ever look at what it might have been, like how much seed there was in the first place? Is it, if it's reducing the Phragmites, the viability of the Phragmites seed bank, is it also reducing the viability? But I guess the fact that you're going to try and overseed means that yes, it does. When I was asking the question, you hadn't talked about, increasing like that's the need for seed restoration, is that all the seeds get damaged by that spray.
Yeah, so the spray doesn't actually affect the seeds themselves. It just kind of reduces the incoming of the seeds, because when the plant is dead, obviously the seeds die as well from the living plant, but the seeds in the seed bank are not necessarily affected. It's just that the frag seeds don't have the long-term viability as opposed to some of the native seeds that would have that long-term viability. So even though there's standing frag that is alive, those native seeds are still persisting in the seed bank from long ago. So those seeds are still present in the seed bank, still persisting, but the Phragmites seeds are not necessarily persisting as long.
Yeah, but I guess the other thing is that there's always like a lot of seed source from the surrounding areas.
Exactly. Yeah.
Awesome. Thanks for the clarification. A question from Kurt. How far apart were your seed bank collections? They've noted that there's a high variability in Phragmites seed viability depending on location collection.
Yeah, so we collected seed bank from the provincial park as well as Big Creek, which is nearby. We tried to get them from as far away as we could, but we did collect them from our existing plots for the most part. So we did keep that in mind, but yeah, it's tough when you're trudging through frag to kind of get them from far apart.
Just a comment that people love the marsh organs. So they sound like a nightmare, but they're really cool. This you might want to go back to one of your slides. Do you know what the insects were that was on the nightshade in one of your photos?
I'm not a bug person, but if anyone does have an idea, let me know. They might be midges. I'm not 100% sure, but I do remember there being a lot of midges in there.
Yeah, they look like midges because they got the feathery antenna, but I couldn't zoom in on them.
Cool. Okay, thank you. Lots of great well dones.
Thank you.
How do you know the plants in your marsh organ came from the seed bank rather than the surrounding area, which you kind of addressed?
Yeah, it's kind of tough. I think if I had unlimited resources, I would have used sediment that was totally free of seeds and stuff, just to make sure that generally those seeds were coming from the seed bank that we added. However, we did put the marsh organs in the areas where we got the seed bank from. So the control marsh organs were in areas of highly dense frag, etc. So there was no seed rain from frag going into reference marsh organs and things like that. But of course, with any experiment, you can't be 100% sure of where the seeds are coming from, but we did try our best to kind of keep control with control, reference with reference, and treated with treated.
Great. A question from Robert. How do you think this works, or how do you think this work applies to drier sites, more along the lines of a wet meadow transitioning into shallow emergent zones?
It's a good question. Jersey's work is working a bit more with marsh meadow, so that might be a question for her if you would like to ask
maybe
her
once
more.
she gets some data, we'll have her for a follow-up.
Yes,
webinar.
100%. This is her second year, so she has 1 year of data already, so she'd be a great choice.
Was there any evidence of the loosestrife beetles when the loosestrife returned? Did you guys see any out there?
Again, I'm not a bug person, so I didn't.
You're I'm not looking for that.
I'm we saw enough bugs, so I wasn't necessarily looking for them, but that doesn't mean that they weren't there.
Another question, given the frag seeds disperse widely with the wind rather than fall from the plant, what explains the higher seed counts in the untreated stands?
Ooh. So I don't know if whoever said it wants to clarify, but do you mean like Phragmites seeds specifically?
I'm guess if you're still on
Yeah,
the call, you
that
can
would be
clarify.
Phragmites seeds. Yeah.
Okay, could you repeat the question just so I can?
Well, just I think you had shown that the density of Phragmites seeds was higher in untreated areas than
Mhm.
others. And given, it's a snowstorm when they're shedding seeds, I mean, they're blowing everywhere.
Yeah.
They're not falling, down onto that sediment in that plot, perhaps. You'd think maybe they would be even falling more on open areas. But
Mhm.
I just curious, again, why there be more in that case. Is it filtering them out somewhat because of the structure of the culms, combing them out of the air?
Yeah, I'm not 100% sure. It's a good question. Maybe I'll have to think about that one.
Mhm. Just a comment from Robert that a forage analysis of dried frog bit compares favorably to alfalfa. So if anyone was,
Oh.
yeah. I don't know if that's another question was some of the regrowth on the Marsh master treated sites due to missed areas and or running down the culms before they are treated.
Yeah, I'm not a total expert when it comes to Marsh master and application, just because that's not necessarily our job, but there, like I said, we're not 100% sure as to why the ground application was a little bit less effective than the aerial application at first. I'm sure there could be many reasons why, but yeah, that's totally a good idea. Yeah.
A question from Lisa. I'm not familiar with the ground treatment and was not expecting to see that big machine, the Marsh Master.
Yeah.
Does it drive through the wetland and spray a large swatch? Or could you just explain like how the Marsh Master treatment was used?
Yeah, so again, we weren't the ones actually doing the treatment, and I've never been in the marsh. I know some of my coworkers have been in close to the marsh when they have been treated, but yeah, it's basically like a big truck that you run through the marsh. It's got those big treads, so it kind of just easily goes through the marsh. And then they spray from Marsh Master itself. So I think it's kind of probably what you're imagining. Yeah.
I'm also not an expert. I can't help. They're so cool though.
So I can talk a bit about that because it's actually my husband that does the spraying.
Oh, awesome.
Yeah, so the Marsh Master is an amphibious machine. So it has pontoons inside of the tracks to help it float. And it is difficult. It gets stuck like going through the channels, and you can't really see anything when the frag is like 12 feet tall or whatever. So it's not an easy job. So they have fashioned a crow's nest on top of the Marsh Master.
Okay.
So we have one person driving, essentially just following a compass, because that's all they can see is a compass. And then the fellow on top is radioing back and forth to say which way to go. Yeah, and so he is just spraying with a handheld gun, hand-directed like spray gun. And then they have to always be in mind of the wind speed. But yeah, they do their best to track with GPS so that they're getting a nice overlap and they're not spraying any native frag. It's pretty involved.
Well, thank you. I'm glad we had an expert on the on the call. Oh, I think there was one more catch when they came in. From Robert, just for clarification, it looks like some of the native seed persistence in the seed bank is substantially higher than the persistence of Phragmites seeds. Is that correct? Okay.
Yeah.
And then on a related note, also from Robert, can you comment on seed viability for Phragmites? At one point, it was considered very low. Do you think, yeah, I guess this is a question that everyone always has, like, wants to know is like, how viable are the seeds?
Yeah.
Did you gain any insight on that?
I mean, I think like my work shows that they're not totally viable, like, because they're kind of reduced in viability like over 1 year with the flooding and stuff. So I think obviously like the main source of control is like killing the plants themselves, but the seeds are also something that we do have to keep in mind because they obviously do remain viable for a certain amount of time, and especially under the falling water levels, that's something that we really have to keep in mind.
Yeah. Okay, I don't see any more questions, but
Okay.
I put up her email, so I'm sure if you think of something, and Megan would be happy to answer them or get in contact with, you know, more information. So just one last, this is recorded, so if you wanted to share, if you like wanted to rewatch it or share this out with colleagues, I will send the recording out to the Eventbrite invitation. It will also be posted up on our YouTube page and our website, so look for that. And then just one final pitch that we do have a April webinar coming up. And speaking of, you guys had so many questions about frogbit, we're going to have the European Frogbit Collaborative give a presentation about like secondary invasion and like what to be on the lookout, you know, for frag managers. So save the date, April 25th. It should be really interesting. And yeah, just thank you all. We love giving these webinars. So thank you, Megan, for presenting. Thank you everyone for attending. And see you at our next one.